Copper. Copper is the third most biologically important transition metal after iron and zinc. Human beings need around 8 mg of copper daily. A deficiency of copper than the optimum value results in the inability of animals to use iron stored in the liver. As a result, the animals suffer from anemia. The copper is bound to proteins in the body either by metalloproteins or as enzymes. Excess of copper is highly poisonous, particularly to fish. This is the reason it is always advised not to throw copper coins in the fish pools for good luck. Human beings can excrete any excess of copper, but a biological defect can result in copper accumulation in the liver, kidneys, and brains. The illness caused by the excess of copper is called Wilson disease. It is hereditary shortage of Curuloplasmin causing copper accumulation in the liver, kidneys, and brain. Patients suffering from Wilson disease are cured by administering ethylene diamine tetracetic acid (EDTA), which form soluble copper complex. The soluble copper complex can be excreted harmlessly through urine. At the same time, many other essential metals involved in other enzymatic reactions from EDTA complex are excreted. Thus, the EDTA treatment can upset many unwanted enzyme reactions. As a result, utmost care should be taken in monitoring the Wilson's disease. Hemocyanin is a copper containing protein which is important as an oxygen carrier in some invertebrate animals. Despite its name, it is non hemoprotein. It is found in the blood of snails, crabs, lobsters, octopus, and scorpions. The oxygenated hemocyanins are blue colored unlike human blood and have one dioxygen molecule attached to two copper atoms. Deoxygenated hemocyanin contains Cu1 and is also blue. Selenium. Selenium is essential to health. All classes of organisms metabolize selenium. In humans and other mammals, circus disease arise from either excessive or insufficient dietary selenium. The toxic effects of selenium have long been known, particularly for grazing animals. In soils with high selenium content, some plants accumulate large amounts of selenium as methyl selenocysteine in order to keep selenium away from the metabolic machinery. Animals that graze these selenium accumulating plants develop severe toxic reactions, including blind, staggers, and alkali disease. Selenium poisoning in human is rare but has occurred where mines have caused selenium to be leached from soil into drinking water supplies. Loss of hair and nails is seen in case of human selenium toxicity, followed by anemia and jaundice. Although selenium is toxic at high levels, selenium is an essential micronutrient for mammalian species. Selenium deficiency in humans results in degenerative condition of the heart tissue known as Kishan disease. This disease is named after a country in China. The accepted minimum daily requirement of selenium for adult humans in is 70 micrograms. Many types of food provide selenium, and particularly see food, meats, and grains and onion family. Mammals and birds require selenium for production of the enzyme glutathione peroxidase. Hydrogen peroxidase and organic peroxides, which cause oxidative damage to cellular components, are enzymatically destroyed by this selenoprotein. Such oxidant activity suggests that glutathione peroxidase, therefore, selenium protects against oxidation induced cancers. Chromium and molybdenum. Trace amount of chromium and molybdenum are necessary in the diet of mammals. Chromium and insulin are both involved in maintaining the correct levels of blood glucose. Molybdenum is the most biologically important member of the chromium family. It is the heaviest element to have a wide range of functions in a living organisms. The most important molybdenum enzyme is nitrogenase. Thus, nitrogenase contains two proteins, that is molybdenopterodoxin and azoferodoxin. Molybdenopterodoxin is air sensitive and brown color species containing 2 MO atoms, 24 to 36 iron atoms, 24 to 36 sulfur atoms together with a protein. It has a molecular weight of about 2,25,000. Azoferodoxin is yellow, air sensitive, and is a derivative of forodoxin. It has molecular weight in the range of 50,000 to 70,000. The enzyme nitrogenase occurs in bacteria and reduces the inert dinitrogen of the atmosphere to the ammonium ion. This reaction is used in the synthesis of protein by the plant. Let us have an idea of the importance of this enzyme by the typical example. The amount of dinitrogen fixed biologically is estimated to be 175 million tons per year, compared with the total of 110 million tons of NH3 produced by the Haber-Bosch process and the distillation of coal. Why is metal as rare as molybdenum so biologically important? There are several reasons to address this question. First, the molybdenate ion has a high aqueous solubility at near neutral pH values, making it easily transferable by biological fluids. The ion has a negative charge, making it more suitable for different environments that are the cations of period 4 transition metals. The element has a wide range of oxidation state whose redox potential overlaps with those of biological systems. Being a soft acid, molybdenum shows a strong bonding preference for the soft Lewis base sulfide. Another important component of these enzymes, nickel. The biochemistry of nickel is poorly understood of all those of period 4 transition element. It is present in some enzyme systems in the form of porphyrin type complex. Some of the tropical trees concentrated nickel to such an extent that it makes up approx 15% of the dry mass. It is highly toxic to most plants and moderately toxic to mammals. Cadmium is a toxic element that is present in our foodstuffs and is normally ingested at levels that are close to the maximum safe level. Kidney is the organ most susceptible to cadmium. Cadmium ingested 200 ppm cause severe damage. Cigarette smokers absorb significant level of cadmium from tobacco smoke. Exposure to cadmium from industrial sources is a major. Use of nickel cadmium battery is becoming a major waste disposal problem. Many reputed companies now accept return of used nickel cadmium batteries so that cadmium metal can be safely recycled. Mercury is a hazardous because of its relatively high vapor pressure. The mercury vapor is absorbed in the lungs, dissolved in the blood, and is then carried to the brain. This causes irreversible damage to the central nervous system. It is slightly soluble in water because of weak metal bonding. Inorganic compounds of mercury are less toxic as they have less solubility in water. Arsenic. Arsenic is an element essential to life. However, we need ultra trace amount of this element. Anything more than a tiny amount causes arsenic poisoning. Arsenic is measurable quantities cause death through enzyme inhibition. Non-metals. The non-metals among the essential elements are hydrogen, carbon, oxygen, nitrogen, phosphorus, sulfur, and chlorine and iodine. Majority of these are needed in trace amounts except carbon, hydrogen, nitrogen, and oxygen, which are abundantly available and constitute most of the chemicals in the living system. Phosphorus. It is an essential element for life. It constitutes an important part of energy rich molecule known as ATP. It is also an important component of bone as appetite, that is calcium hydroxide phosphate. Partial formation of fluoroapatite strengthens the structure of the teeth and makes it less soluble in the acid formed for fermenting organic materials in the teeth. Iodine. Nearly 70% of the iodine present in the human body is located at one place, that is the thyroid gland. It is used in the synthesis of hormones thyroxine and triiodothyronine. These hormones are essential for growth, for the regulation of neuromuscular functioning, and for the maintenance of male and female reproductive functions. One common cause of goiter is lack of iodine in the diet. A symptom of goiter is a swollen lower part of the neck. The enlarged is the attempt of a thyroid gland to maximize absorption of iodine in iodine deficient circumstances. In order to remove deficiency, potassium iodide is added to the common household salt, such a salt is known as iodized chloride ion. The chloride ion has a vital role in the ion balance of our bodies. It does not appear as to play an active role, but simply act as balance the positive ions of sodium and potassium. It is a major ion found inside the cell. It is essential as an initial electrolyte for higher mammals and plants. Chloride ion is essential in digestive juices. Deficiency of chloride ion in infants lead to impaired growth. Nearly 30 elements are now recognized as essential elements for at least some forms of life. It includes most of the elements through atomic number 35 except from B, E, A, L, S, C, T, I, G, A, G, E. Along with noble gases, molybdenum and iodine are also essential. It is interesting to note that all elements appreciably abundant in human body, with the exception of phosphorus, are also abundant in seawater, suggesting without doubt that our family tree has root in the sea. Metalloporphyrins. Porphyrins constitute an important class of bioinorganic compounds in which a metal ion is surrounded by 4 nitrogen of a porphyrin ring. Porphyrins are macrocyclic tetradentate ligand made up of 4 pyrrole ring linked together through methane bridges. This tetradentate ligand has a conjugated double bond system with 4 nitrogen donor sites. Two of the 4 nitrogen are tertiary nitrogen, while the remaining 2 nitrogens are secondary nitrogen. The tertiary nitrogen atoms form coordinate bond by donating a pair of electrons each on the each and form a covalent bond with a metal ion as shown. Thus, porphyrin ring serves as a tetradentate negative ligand. As a result, di-positive cations like Fe2 positive, Ni2 positive, and Mg2 positive will form a neutral complex with porphyrin, which in the process loses 2 protons. When the 4 pyrrole rings of a porphyrin are substituted by organic group together other than hydrogen, it is called a porphyrin. Thus, porphyrins are macrocyclic tetrapyrrole system with conjugated double bonds. Together with various groups substituted at the periphery of the ring, the substituent attached to the ring are usually organic groups with electron withdrawing or electron releasing characteristics. The complex formed by the porphyrins with the metal ions are called metalloporphyrins. These are biologically important compounds and play a very important role in the animal and plant kingdoms. Porphyrin ring system is fairly rigid because of the delocalization of electrons in the molecules. Although the skeletal structure is fairly rigid, significant variations have been observed among the metalloporphyrins in the interatomic distance between the pyrrole nitrogen and the metal ion at the center, determining the size of the central hole. From a knowledge of the covalent bond radii, one can estimate that a bond between nitrogen atom and an atom of the first transition series should be about 2 angstrom long. In brief, an ideal size of an atom to fit in the hole is 2 angstrom. The size of the hole in the center of the porphyrin ring is ideal for accommodating metal ions of first transition series or lighter alkaline earth metals. A metal ion of appropriate size sitting at the center of the porphyrin ring will be surrounded by four nitrogens of the pyrroles and forming a square planar geometry. It means that the metal ion and the four nitrogen lie in the same plane. However, when the size of the metal ion is smaller than ideal, the ring will be reluctant to accommodate precisely the metal ion. In this way, the ring will either re- readjust or distort itself in order to accommodate the metal ion. On the other hand, if the size of the metal ion is larger than ideal, the transition metal ion cannot fit in the hole and stays out of the plane of the four nitrogen atoms. It means that the metal atom sits above the plane of four nitrogen atom and requires a square pyramidal geometry. A nature of metalloporphyrins vary with the nature of the metal ion and that of the organic groups attached at the eighth position of the four chiral ring around the periphery of porphyrins. A group of related compounds, the phthalocyanins, are isoelectric with the porphyrins. These compounds are of interest balance of their intensely colored metal complexes, which are of utmost commercial applications in dyes and pigments. The size of the hole in these phthalocyanins ring is about 0.1 angstrom smaller than in porphyrins as a result of nitrogen atom compared with carbon. Compare the covalent radii of carbon and nitrogen. Two of the most important metal complexes of porphyrins are chlorophyll and heme, which are being discussed. Chlorophyll is a magnesium complex of porphyrin which plays an important role in photosynthesis. Photosynthesis is a remarkable redox reaction in nature that convert water and carbon dioxide into sugar and oxygen in the presence of sunlight. 6CO2 plus 6H2O gives you C6H12O6 plus 6O2. The process is the primary source of food that we eat and the molecular oxygen that we breathe. The function of chlorophyll molecule is to absorb photons in the red part of the visible spectrum and pass this energy of excitation to other species in the reaction chain. The ability to absorb the light is due to conjugated Pauline structure of the porphyrin ring system. The role of magnesium is to make the entire molecule rigid so that energy absorbed is not easily lost. There are a number of similar but not identical chlorophyll molecules. It is important to note in chlorophyll A, the basic porphyrin system has been modified in two ways. In pyrrole ring, one of the double bonds has been transhydrogenated to form a magnesium dihydride porphyrin complex. In addition, a cyclopentanone ring has been fused to the side of the pyrrole ring as shown. In brief, absorption of sunlight by chlorophyll is the first step in the conversion of sunlight into usable energy. It will not be out of place to say that chlorophylls are nature's solar energy collectors. Heme is an iron porphyrin which is present in hemoglobin and myoglobin, which is being discussed.